Welcome to my channel, The Wise Pathogen. Today we will be discussing about the history and development of microbiology. But before that, we need to understand the concept of microbiology. As we are able to see on screen, that microbiology is the study of microscopic living organisms such as bacteria, fungi, algae, protozoa, and the infectious agents at the borderline of life called viruses. Now, you must be wondering why viruses are not classified as living organisms. The reason being that viruses do not possess cellular machinery for metabolism, which is the defining feature of living organisms. Viruses are also not able to reproduce independently and depend on host cells. It concerns their form, structure, reproduction, physiology, metabolism, and classification. It also covers the following points: the distribution in nature, relationship to each other and other living organisms, effects on human beings and other animals and plants, physical and chemical alterations in the environment, response to physical and chemical changes. Today, we are aware of the concept of microbiology. However, there was a time when people were not acquainted with the idea of microorganisms. Now, we are going to dive into the contributions of the scientists that led to the development of microbiology. Scientists started researching about microbiology in the late 19th century. Till the 1880s, people thought that diseases were a result of sins. Different cultures had different beliefs. The birth of microbiology began when people began to grind lenses from pieces of glass and combine them to produce magnifications great enough to be able to observe microbes. In the 13th century, Roger Bacon thought diseases are caused by invisible living creatures. Girolamo Fracastoro and Anton van Plenkes made the same suggestion with no proof. In the year 1658, a monk named Athanasius Kircher suggested that worms are present in decaying bodies, meat, milk, and diarrheal secretions that are not visible to the naked eye. Despite no proof and descriptions to the claim, he was the first person to recognize the significance of bacteria and other microbes in diseases. Robert Hooke was a British scientist known for his discoveries spanning physics, astronomy, and paleontology. He also coined the term cell and was the first to publish a depiction of a microorganism in his book Micrographia. His work paved the way for others to keep innovating in the field of microbiology. Now we are going to talk about Antony van Leeuwenhoek. Antony van Leeuwenhoek was the first person to observe and accurately describe microorganisms, that is, bacteria and protozoa, called animalcules in 1676. He was the first to report his observations with accurate descriptions and drawings. He was a Dutch linen merchant. However, he spent much of his spare time constructing simple microscopes composed of double convex lenses held between two silver plates. During his lifetime, he made more than 250 microscopes consisting of home-ground lenses mounted in brass and silver, out of which the most powerful would magnify about 200 to 300 times. These microscopes slightly resembles the current day compound light microscope that can magnify about 1000 to 3000 times. His descriptions of protozoa were so accurate that many of the forms that he described are easily recognized today. He carefully recorded his observations in a series of letters to the British Royal Society. In one of the first letters dated September 7, 1674, addressed to Henry Oldenburg, Secretary of the Royal Society, he described the very little animalcules which we recognize as free-living protozoa. On October 9, 1676, he wrote, quote, "In the year 1675, I discovered living creatures in rainwater which had stood but a few days in a new earthen pot, glazed blue within. This invited me to view this water with great attention, especially those little animals appearing to me 10,000 times less than in those which may be perceived in the water with the naked eye. He described his little animals in great detail, leaving little doubt that he saw bacteria, fungi, and many forms of protozoa. For example, he reported that on June 16, 1675, while examining well water into which he had put a whole pepper the day before, I discovered in a tiny drop of water incredibly many very little animalcules, and these of diverse sorts and sizes. They moved with bendings as an eel always swims with its head in front and never tail first, yet these animalcules swam as well backwards as forwards, though their motion was very slow. His letters were read with interest by the British scientists, but the importance of his discoveries evidently went unappreciated. Now we are going to discuss about the differences of spontaneous generation and biogenesis. Now the concept. Spontaneous generation is the idea that life can arise spontaneously from non-living matter. For example, it's stated that maggots can rise from rotting meat or mice could emerge from a mix of dirty clothes and wheat. On the other hand, biogenesis states that life can only come from pre-existing living organisms. Simply put, all life arises from life-like bacteria multiplying from other bacteria. Now the historical background. Spontaneous generation was widely accepted for centuries, all the way until the 17th century. Back then, people did not have the tools or knowledge to observe microscopic life or test these claims. However, by the 19th century, advances in science and some clever experiments led to the acceptance of biogenesis as the true explanation of how life originates. Now, the key supporters. Spontaneous generations had major historical figures backing it, like Aristotle, who proposed it in ancient times, and later scientists like John Needham, who tried to support it with experiments. Meanwhile, biogenesis was supported by scientists like Francesco Redi, who debunked spontaneous generation with his maggot experiments, Lazzaro Spallanzani, who showed microorganisms come from existing microbes, and the legendary Louis Pasteur, whose swan-neck flask experiment and John Tyndall's dust-free chamber experiment put the final nail in the coffin for spontaneous generation. Now the examples proposed. Proponents of spontaneous generation cited examples like maggots appearing on rotting meat or mice forming from dirty clothes. To them, these observations seem to confirm that life could emerge from non-living matter. In contrast, biogenesis proved that life forms like bacteria or maggots only arise when pre-existing organisms are involved. For instance, flies laying eggs or meat or bacteria growing from other bacteria. Now the scientific status. Today, spontaneous generation is discredited. It is seen as a historical belief that has no scientific basis. Meanwhile, biogenesis is fully accepted as a fundamental principle in biology. It has been supported by experiments and forms the foundation of modern science. Spontaneous generation does not play a role in modern science. It is a relic of history that reminds us of how far science has come. Biogenesis, however, is a cornerstone of modern biology. It is crucial to our understanding of reproduction, heredity, and microbial life, and it paved the breakthroughs like cell theory and germ theory. To sum it up, spontaneous generation and biogenesis represent a major shift in scientific thought from outdated beliefs to evidence-based understanding. Now we are going to understand the series of experiments or informations that led to the discrediting of the spontaneous generation. Aristotle formulated the hypothesis based on earlier contributions from natural philosophers, and it remained widely accepted for nearly 2000 years. The theory was challenged by Francesco Redi and Lazzaro Spallanzani in the 17th and 18th century. However, spontaneous generation was still not proven wrong. The view was finally challenged by the Italian physician Francesco Redi, who carried out a series of experiments on decaying meat and its ability to produce maggots spontaneously. Now, what Redi did here was he took 3 glass jars and in which he put three decaying meats. The first jar was uncovered. Second was covered with a gauze. And third was sealed. Now, in the first jar, since it was uncovered, flies were attracted to it. They laid eggs. As a result, maggots formed. Now, in the second case, the flies were attracted to the jar, but it was not able to enter the jar. So, what they did was, they were attracted to the gauze, they laid eggs on the gauze, and the maggots formed on the gauze. And in the case of the third jar, since it was completely sealed, no flies were attracted. Thus, the generation of maggots by decaying meat resulted from the presence of fly eggs, and meat did not spontaneously generate maggots as previously believed. In 1748, the English priest John Needham reported the results of his experiments on spontaneous generation. Needham boiled mutton broth in a flask, which he tightly stoppered. Eventually, the flask became cloudy and contained microorganisms. He thought that organic matter contained a vital force that could confer the properties of life on non-living matter. However, the scenario was the boiling was not long enough to kill the microorganisms. Now, this experiment was done by Lazzaro Spallanzani. He wanted to improve the experimental setup done by John Needham. So what he did was, he boiled beef broth. This is beef broth. He boiled beef broth for an hour and then sealed the flask. No microbes appeared following incubation, but his results, confirmed in repeated experimentations, failed to convince John Needham, who insisted that air was essential to the spontaneous production of microscopic beings and that it had been excluded from the flasks by sealing them. This argument was answered some 60 or 70 years later independently by two other investigators, Franz Schulz and Theodor Schwann. Schulz passed air through strong acid solutions into boiled infusions, whereas Schwann passed air into his flask through red-hot tubes. In neither case did microbes appear, but the hard-die advocates of spontaneous generation were still not convinced. Acid and heat altered the air so that it would not support growth, they said. About 1850, H. Schroeder and T. von Dusch performed a more convincing experiment by passing air through cotton into flasks containing heated broth. Thus, the microbes were filtered out of the air by the cotton fibers so that growth did not occur, and a basic technique of plugging bacterial culture tubes with cotton stoppers was initiated. The concept of spontaneous generation was revived for the last time by Felix Pauchet, who published in 1859 an extensive report proving its occurrence. But he reckoned without the ingenious, tireless, and stubborn Pasteur. Irritated by his logic and data, Pasteur performed experiments that ended the argument for all time. Pasteur first filtered air through cotton and found that objects resembling plant spores had been trapped. If a piece of the cotton was placed in sterile medium after air had been filtered through it, microbial growth occurred. Next, he placed nutrient solution in flasks, heated their necks in a flame, and drew them out in a variety of curves. The swan-neck flasks that he produced in this way had necks open to the atmosphere. Pasteur then boiled the solutions for a few minutes and allowed them to cool. No growth took place even though the contents of the flasks were exposed to the air. Pasteur pointed out that growth did not occur because dust and germs had been trapped on the walls of the curved necks. If the necks were broken, growth commenced immediately. Pasteur had not only resolved the controversy by 1861 but also had shown how to keep solutions sterile. Finally, John Tyndall conducted experiments in a specially designed box to prove that dust carried the germs. He demonstrated that if no dust was present, sterile broth remained free of microbial growth for indefinite periods.